Good day, this is Professor Will, CBMD PhD. Today's Friday, July 2nd, 2021. It is 12:41 and 36 seconds a.m. Eastern Standard Time. Today's research, multi-talented filaments. And don't forget to check out my website. So let's hop to it here. Multi-talented filaments in living cells. Researchers investigate special properties of protein structures. Date July 1st, 2021. Source, University of Göttingen. Living cells are constantly exposed to a wide variety of mechanical stresses. Heart and lungs must withstand expansion and contraction. Our skin must be resistant to tearing whilst retaining elasticity. Immune cells are squashy, so they can move through the body. Intermediate filaments, special protein structures, play an important role here. Researchers have now measured what determines the properties of individual filaments, and which features only occur through the interaction of filaments and networks. One of the most important systems that cells use to ensure their stability, elasticity, and resistance to mechanical stress is the cytoskeleton, an intricate network of proteins and filaments, predominantly formed by three types of thread-like protein structures, each of which has different functions and properties. The intermediate filaments belong to this group of protein structures, and these filaments form networks that can be subjected to a tremendous force without being damaged. They are the shock absorbers of the cells. At the same time, these intermediate filaments can serve as an inner tether in the case of powerful deformation, and this can prevent the cell from being torn apart. And to investigate these properties, the researchers created artificial networks of intermediate filaments in the laboratory and use the movement of small embedded spheres to study how the entire network behaves. In fact, there are overlapping effects in the networks, the stretching behavior of the individual filaments, and the force and frequency with which the filaments interact at points where they cross intersections. For this reason, the researchers investigated these aspects separately by first stretching individual filaments to determine the force required for the stretching. They then brought two of the filaments into contact with each other in a crisscross pattern and pulled on the point of intersection by moving one of the filaments. By arranging the filaments as if they were a microscopic violin, they were able to determine the exact strength and frequency with which the filaments bind together. They were also able to replicate these results with computer simulations. In addition, the team observed that the networks transform over an unexpectedly long period of time and slowly age over the course of a week. Because the filaments become longer and longer, or join together to form bundles. All these observations increase our understanding of how our cells manage to be so incredibly robust and yet flexible. In addition, a clear picture of intermediate filaments helps us to understand how and why the mechanical properties of cells change. For example, during wound healing or in metastasizing cancer cells. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.